very much. This little talk on what used to be Pipridae, then Pipra, and now Pseudopipra is a result of the work of this team, especially Jacob Berth, who was my undergraduate at Yale and now a grad student in Irby's lab at Cornell, soon to be starting a postdoc at Michigan. About a week ago, Chris Balakrishnan said, "Hey, can you present some of Jake's stuff at this meeting? Because neither of them could make it." And I was a great pleasure to do so. This is well, an understudied mannequin. It's an underwhelming bird in the field in many cases, and yet those of you who have seen it in the field have been aware for a while of a stunning vocal diversity in this bird. It ranges from Central America in montane Costa Rica down through the Andes, and then in Amazon, and then with an isolated population a little bit further north of us down to about Rio. So in order to analyze that, I've actually been dreaming of this project since about 1988, and the good thing is that when you don't get things done, methods get better, and then the students get smarter than you are, and before you know it, you got like some great breakthroughs happening. So this is fantastic work by Jake. What we did was this is a quick overview, we used data from 277 individuals from Costa Rica to Rio. I'll present results of RADseq analysis of 232. We also, before the RADseq was available, did a preliminary NV2 analysis for 160 individuals, 80 locations, and 10 of the 13 subspecies within the group. Again, we did RADseq and some minor NV2. We also present a vocal analysis. We have recordings from 198 individuals from all 13 subspecies represented. Jake is an analytical animal. He got to rip the data out of his hands because he really likes to chew it up. And so he's done an enormous number of stuff, both including population genetic work, phylogenetic inference, and landscape genetics, and then some vocalizations. The collections of these tissues could not have been possible without loans from many museums. And notable, of course, in my experience, I've never had work that used so much tissue curated in South America. So thank you to all these Brazilian institutions here at IPA and Museu de Zoologia in Sao Paulo and others, Rio as well. Our collection localities, this is not as clear as I like, but we include Central America, throughout Amazonia, southeastern Mata Atlântica, and then in the Andes. Unfortunately, very hard and still to get access to collections in the northern Andes of Colombia, and also critical historical material from Ecuador was unavailable for the RADseq because of a freezer meltdown. So that's all represented here with our set of sampling. The phylogenetic analysis of this was extremely clear. This is a very diverse group. I'll try to start. I have another few slides to get this across. This looks like this radiation is out of the Andes. Pseudopipra or Pseudopipra is thought to be the sister group to Ceratopipra, primarily or initially lowland radiation. So this is probably the original montane sister group to Ceratopipra. The first division is deep down in the tree, down here, is between the northern Andes and the Central American population and the southern Andes. This happens about right in the area of the Huallaga River Valley. And then the Central America sister group to the northern Andes, which we have represented unfortunately by only one sample. But then this southern Andean clade is also diverse, including at least the two well-differentiated forms. And then this whole lowland radiation has a single ancestor or a single descendant with a montane ancestor. There is a radiation in southern Amazonia, including Atlantic forest, and then the Guiana Highlands, radiation, and then this very interesting Western Napo population with complex history. When we do a structure analysis, the structure informative, the partitions get maximally informative about 5. We think there's a way more diversity, and part of that is just the very patchiness of our sampling. So, for example, we have this northern Andean clade represented, unfortunately, by a single phylogenetically very distinct bird. So let's look at this projected onto the landscape. Here again is our ancestor. We have the northern Andean clade, which is the sister to the right. That's from about the Huallaga River, or the west bank of the Huallaga River, up through here. These again underrepresented, but with this in this clade. Then this combination of southern Peruvian groups with both phylogenetic and population genetic identity, and then this lowland radiation that includes a southern Amazonian clade with a series of successive relationships, just like the Congo with many other population genetic or many other phylogenetic things, say trumpeters, Sophia, etc. And then a Guyana group with a good ability to differentiate the Suriname and Western Guyana areas. Interestingly, this one population, Pygmaea, was not, the genes were, or the sample was not large enough for us to include in the RADSEQ, but it is a very distinct population right down there in about the mouth of the Rio, the Huallaga River Valley. And it appears to be on the basis, shown here in red, on the basis of mitochondrial genome DNA, is the sister group to the entire lowland fauna. Other fascinating things here in our RADSEQ analysis, this upper Napo, or this Napo population here, has a very odd combination of sharing identity with the Guyana Shield population and the southern Amazonian population. Jake hammered the hell out of this problem, you'd have to say, with lots of analysis. The bottom line is that this Napo population seems to be phylogenetically sister to the rest of the southern Amazonian clade, but has a huge amount of introgression from the western, or from the Guyana clade. There's also, so here is PC1 and 2. This is the Napo population, showing that it is more similar to the Guyana clade, but has a whole dimension that's different from either the lowland or southern thing. So this is not a case of hybrid speciation, I don't think. I think it was a distinct lineage at the time, prior to this integration, but that has occurred. So with this enormous amount of data, you learn lots of new stuff. Yeah. He's also looked at things like estimates of migration rate, other kind of population genetic stuff. I'm much more interested in the vocalizations, so I'll show a little bit of that. I couldn't, in this short amount of time, get this slide from Keynote into this. Here we go. Here are a sample of vocalizations from across the species. Let's see. Yeah, got that? Okay, that's one. And I don't know why it's in this order, but that's Teresa Theo, my student. These are all different vocalizations by males on the left. So these are, I think everyone would admit, really different. Not surprisingly, when we do math, we can show that too. And so there's also, we found another call type, which is not an adverse vocalization, that was also distinct and associated with clades. So here is the PC analysis, PC1 and 2 versus those 10 or 11 different male lab types, and again, 2 versus 3, and their spatial distribution, and then the call types. So bottom line, there are lots of species of Diazythia or Pseudopipra. This is a conservative breakup where we recognize 8 different species. These are including the northern Andean clade as the Central American species. This is a bunch of the Andean forms. And here's the southern Andean form, including lots of populations that don't have names, but are distinct. And we didn't, we may have, but we lack vocalizations for them. And then the Southeast Atlantic one. What's the bottom line here? I think there's a lot of underappreciated diversity. I think that we need to add some of this primary history and population studies to the study of behavior. So thank you very much for your time.